Hello and welcome to a short introduction of GoMapMan, an application for plant gene functional annotations. The center of the application is the so-called ontology tree view, which displays the MapMan ontology, which is an ontology organized in a tree-like structure. You can move deeper through the ontology by clicking on various so-called bins, which describe these functions. The deeper that you go, the more specific the annotations will become. For instance, here I moved deeper into the tree of stress biotic receptors. This one also has two additional sub-bins, but they are not of interest at the current time. Each selected bin will display the genes and the ortholog groups which are described with that particular functional annotation. Here we can see Arabidopsis, potato and rice genes. The first thing that one can do is unselect various plants. Let's say we don't want to look at rice anymore, we just unclick it and wait a little bit for it to load. Once it has loaded, we can see that there are no more pesky rice genes anymore. The other thing that also can be done is select various different ortholog types from the list, which I will do now. And again, wait for it to load. As you can see, the current display is slightly different from the previous one, which is the case for all different ortholog types selected. And these changes actually reflect the differences between different ortholog groups, which are the result of different algorithms which were used to get them. Clicking on an ortholog identifier will open up the ortholog details page, which will contain various bits of information. Most importantly, which bins the genes of the ortholog are described with, the list of all the genes that belong to the ortholog group with their descriptions, and also some annotations, if they exist, but most commonly for orthologs they will contain identifiers of other plant species which have not been included in GoMapMan yet. If we return to the tree view from the ortholog details view, we can also click on a gene identifier, which will open the gene details page, which is structured very similarly as the ortholog details page. And first you will have some information from the parent database, such as description, short names, synonyms or genomic contexts, and also the date of last modification. Sadly, this gene does not have any short names or synonyms, which is often the case. Then the ontology annotations of this particular gene follow, the ortholog groups in which this gene belongs to, and then transcripts or microarray features which are connected with this gene. This particular one has 3 microarray probes from a potato Agilent microarray design. Lastly, there are also annotations to other external databases, such as other ontologies, gene ontology, or other structure and protein function databases, PFAM, Panther, Interpro, Superfamily. We also connect it to pathway databases such as KEGG or literature links in PubMed, which sadly this gene doesn't have any annotations either. Also, a simple search has been implemented, which allows you to search through genes, orthologs, or microarray features separately. For genes and microarray features, you are able to select which plant species you want to look through. For example, you don't want to look through rice anymore, you unclick it. While for orthologs, the search is species independent. Another one are the exports, which take the information that we have stored on the gene bin connections in the database, and we prepare import files for other software such as gene set enrichment analysis or MapMan tool. We also have 2 exports for BioMine and SEX tools. We have a generic export set and the export of the ontology in the OBO format. Both gene set enrichment analysis and MapMan folders will contain mappings for all the plants and different types. Every plant always has a genome mapping, and some also have transcript mappings, such as the case of beet. Or also alternatively, other plants will have microarray probe mappings, such as Affymetrix and Agilent for the tobacco plant. Moving in the export folder structure will eventually get you to the archive, which contains all the previous mappings for all the 5 selected sections, as we generally try to update these mappings every time a new plant species has been added or a new ortholog type has been added, or the number of manual curations has reached a particular arbitrarily selected threshold. We don't discard the old ones, because that would obviously bring some reproducibility issues. So they are all nicely archived and packed here. The last part would be the manual curation, which will be covered in the second tutorial, so currently thank you for your patience.